well. It's okay?
Yeah.
Okay. So, this is a topic entitled How Molecular Tools Have Helped Me to Interpret the Taxonomy of Some Genera of Amaranthaceae. So first of all, I want to thank Dr. Anil Kumar for his kind invitation to participate today in this international webinar. Thank you to everybody in charge of this meeting organization and to the audience for being interested in this talk presentation. I also want to thank Dr. Arya for scheduling my talk presentation. We had a blackout here yesterday, and it was impossible for me to connect to the webinar. So it's a very exciting experience for me to be for the first time in India. It doesn't matter the distance because we are united by sharing a common passion, which today is called SYSTEMA 2020. So my talk presentation has 9 key points that I hope to reach them successfully in 1.5 hours. I have prepared 2 examples related to my experiences using molecular data on my projects. I hope we have time enough to see them all. So the key points includes a personal introduction. Why is taxonomy alpha a crucial activity? Why is taxonomy alpha not considered a scientific activity by some authors? Which are the most important changes in systematics during the last 70 years? Why is important the homology term? Which are the most followed assumptions to propose putative homologies? The 7th point is homology assessments, how pollen and DNA sequence impact in both taxonomy alpha and phylogenies of Amaranthaceae. The 8th, homology assessments, how ontogeny and DNA sequence impact in both taxonomy alpha and phylogenies of Amaranthaceae. And at the end, I just want to present some very few general conclusions. Okay? So as a personal introduction, well, this is Mexico. Mexico is one of the most biodiverse countries, as well as Brazil and India. Mexico has around 2,000 square kilometers in an area that includes 3,000 species of angiosperms and more than 12 endemics. It's a very rough place. You can see here. So in the east, we have the Sierra Madre Oriental. In the west, we have the Sierra Madre Occidental. As you can see, the ocean winds coming from Atlantic and Pacific leave humidity in the mountains. And by the time that reach the center of the country, they have lost most of the humidity. For that reason, Mexico is a more drier place with a bigger desert formed as the sunken sink, called the Chihuahuan Desert. According to Norman Myers, an international conservation of hotspots designation, one of the most important points is the Mesoamerican area, which is basically a tropical zone. Unfortunately, as it has been mentioned, those points, those hotspots, have been characterized not only for the endemicity and less altered nature, but also for having the major number of population, poorness, and violent and criminal activities. Mexico is not the exception, and as I will explain later, we have to conduct fieldwork in areas where drug leaders are the major danger. I think I have to tell you this because taxonomists have to work sometimes in danger situations that most of the time get an impact in the way we conduct our projects. To be a taxonomist nowadays is a risky job. So all kind of vegetation is present in Mexico. And as you can see, this is the Yucatán Peninsula. It's the only flat plain without mountains, which is the place where I live and I work. So I am here. Well, you are welcome to visit us when the conditions allow it. The Yucatan Peninsula is a great place to be and carry out my job as a taxonomist, particularly here at CICY, my home institution. It's one of the most important scientific places in this part of the country with the graduate and student exchange programs. Now, now that I have introduced myself and my place of origin, I hope you can see that with a huge diversity, we need to make efforts to continue collecting, identifying, and describing our biota. USA biologists have been the ones more participative in this activity in Mexico. Much collaboration has been acquired with USA, and just in recent years, there have been more German biologists' involvement. We say the rich countries have the economic resources, and the middle-income economy countries as Mexico have the biodiversity. We are convinced that we need more taxonomist specialists in different groups in Mexico. So, why taxonomy alpha is a crucial activity? When I think in taxonomy, I always go back to this image that you can see. This is taken from the book entitled Vascular Plant Systematics by Albert Bradford. It was written in 1974. I believe that taxonomy is the central topic enriched by too many areas such as chemistry, anatomy, and too many others as we can see here. It's the main source that helps these areas in the correct identification of the organisms that are the topic of study for ecologists, anatomists, or any other people interested that requires a scientific name, which was provided by taxonomists. Taxonomists that have dedicated years of training, recognizing organisms, and sometimes risking life to collect the material. Then our work is enriched by fieldwork, herbarium, museums, as well as other collections. This is an endemic tree from Mexico. As you can see, the name in Nahuatl, which is one of the ancient languages in Mexico, the meaning of the name is flower in form of hand. So as can you see, there are five stamens that seems like fingers, and that the scientific name is Keratodendron pentadactylum, which refers to this feature. It's like a hand with 5 fingers, right? So this is an endemic species, and this is what taxonomists do, to offer scientific names that try to give in only 2 words the most relevant features of organisms. So the recognition of similarities and differences between organisms is a basic human activity, and in combination with a system of nomenclature is essential for accurate communication. Modern systems of classification attempt to serve 2 needs: to present our current state of knowledge regarding the relationships of organisms, and to provide, in combination with a system of nomenclature, a retrieval system for information about organisms. So why is taxonomy alpha not considered a scientific activity by some authors? It has been mentioned that alpha taxonomy is the discipline that describes, identifies, and gives name for taxa. It has been said that this practice is subjective because the recognition of taxa is based on empirical observations and depends on the experience, the knowledge and ability of the taxonomist. This practice is not scientific because it doesn't use the scientific method. When we propose a new species, for example, the character descriptions has been considered intuitive or subjective. It is true that there are many botanical glossaries, and as botanists, we use a universal language. That means that, for example, the lanceolate form concept is the same for Mexicans, American, Chinese people, but sometimes this variation or characters recognition can be totally wrong. I will give you an example of this matter in a few next slides. Taxonomists that work Samarantaceae are strongly using, are wrongly using some terminologies that shouldn't continue in use. So as you can see in these slides, there are two main scientists. One is on the left side, we have Arthur Cronquist. He offered an important classifications in the plant taxonomy. On the right, we have Willy Henning. So it's important to mention that we have in this slide just showing that there are two different ways of thinking about evolution. For example, it is said that previous taxonomic classifications were ruled by an authoritarianist vision from people that gave direction about the evolved and primitive groups. Then, thanks to William Henning and the cladistic approach based on finding putative homologies and create the method to test homology assessment, then the method was able to be repeated by other scientists, and therefore the taxonomy was no longer more a non-scientific discipline. They were called systematics. And the main reason is because now it's applied a scientific method. That means there are two main steps in this method, which are the hypothesis and to repeat by self or reproduce by other scientists the same conditions and to get the same results. So I'm not saying that taxonomy alpha is unnecessary. On the contrary, you will see along my talk presentation, and I hope I can reach this point, how it's related the alpha taxonomy with many other disciplines. I am saying that each discipline project should have their own scopes and limitations. I just want to emphasize the big impact about that cladistic tools brought on plant systematics. Which are the most important changes in systematics during the last 70 years? As an example of previous classifications, there exists a proposed by Cronquist, mainly based on chemical compounds. So in this slide, you can see that Cronquist, just for the subclass Caryophyllide, he proposed three orders: Polygonales, Plumbaginales, and Caryophyllales. He gave an approach of families and species diversity according to his conception as circumscription of this group. Later on, during the last 70 years, thanks to PCR techniques, the use of DNA sequences, PCR-based markers that replaced allocymas in studies at the population and infraspecific level, the progress in plant systematics was notorious. One attraction of the approach was the potential for constructing phylogenetic trees totally independent of morphological data. Also, the existence of modern computers and software, as well as major capacity of computers, RAM, or memory to run long phylogenetic processes with many data, have also helped to conduct phylogenetic proposals. Then, the main progress in systematics was influenced by cladistics, DNA sequencing, and computers. Therefore, it is not a surprise that researchers with vision and monetary capability from the APG, a group of botanists that named themselves the Angiosperm Phylogeny Group, the IPG, presented an additional classification in 1988 for the angiosperms, in which the orders appear to be monophyletic, based on available data, much of it molecular. This represents an example of circumscribing phyletic groups within the context of a ranked system of nomenclature. Then, in the light of a new philosophy dictating that classification should be based on evolutionary relationships, the phylogenies and the homology concepts were associated. So as you can see, the classification proposed by Cronquist was influenced by an authoritarianist point of view, and once on the light of cladistics and a proper method to use characters and test phylogenies, we can see that on the light of the APG, those groups were recognized as different categories and different relationships. So the previous three orders recognized by Cronquist as orders here are recognized as families by the APG. Why is important the homology term? We can see that homology is a word that already existed before the concept of phylogeny, but now both are very closely related terms. The opposite of homology is the homoplasy, and here are the most recent definitions that I have found. So homology means similarity due to a common origin at the species level. It's the same by phylogenetic origin. Homoplasy, it's phenotypically identical structures that occur on different lineages or clades are homoplastic. It has been argued that homoplasy cannot naturally exist. It is always the result of an error, either in the phylogenetic hypothesis or in the character coding. It has been considered useful to retain the term homoplasy for structures that occur in different clades, but nevertheless pass the common origin test. So it's important to mention that there are many concepts, contexts, and terms around homology. You can see that there is a lot of information and bibliography about homology, and this term hasn't been finally described. There are too many researchers that continue making discussions about homology. One more relevant terminology was suggested by Fitch in the 70s. These are the concepts used for molecular data. However, based on recent papers, these concepts can be used at any kind of data: morphology, genes, ontogeny. So in this slide, you can see that we use morphological data. Homology originally was exclusively described for morphological concepts, and later on become accepted in a phylogenetic context. On the light of the molecular data, 3 types of homology were recognized: orthology, paralogy, and serology. Orthology means the relationship among DNA sequences that diversify through speciation, thus originating from the same ancestral sequence. Paralogy means the relationship of DNA sequences that diverge through gene duplications, refers to duplicates of a same ancestral DNA sequence. Serology is the relationship among DNA sequences that are horizontally transferred, referring to the relation between the DNA of a donor and an acceptor species, which doesn't share common origin. So these three concepts used in molecular data now on recent papers can be also used for morphological, genes, chromosomes, or any other kind of data. Okay, so which are the most followed assumptions to propose putative homologies? We assume that we are using orthologs, which is a kind of an homology. Orthologs means same ontogenetic origin. We shouldn't use analogs, which means different ontogenetic origin. And what's ontogenetic origin? Well, it depends on the level and sublevel of the underlying question. For example, it can be the same position, the same primordium if we are talking about plants, or the same cell lineage if we are talking about animals at the same organ. It can be the same relative position of the tissue at the histological level, the same position of amino acid or a mononucleotide within a sequence at the molecular level. So those are the assumptions that we are using. Of course, we collect a group of data. It can be from pollen, anatomy, trichomes, leaves, molecules that will be evaluated through the homology test, and those that pass these tests have been corroborated as homology. It's very important to establish what's the correct definition of a character and a character state. So what's a corroboration test? So at first glance, when we design a study, there are probably many possible characters and character states that can be hypothesized as homologous. However, as homology tests are applied, the number of features being corroborated as characters or character states is usually reduced. Once we assess homology, we have to use origin instead of development. A character, this is the meaning of a character, are qualities assigned to structures differing from each other by either another origin or by deep divergence. Character states are observable traits of a given character caused by developmental divergence of structures with the same origin. We are going to see some examples on the next slides, and I hope we can reach the concept of character and character states as well as corroboration tests. The next topic is about homology assessments, how pollen and DNA sequence impact in both taxonomy alpha and phylogeny of Amarantaceae. The next slide will show you how I have applied the concepts I have mentioned up to now. You also will see how these concepts can be used in your own research if you find them useful. So this is the group of plants that I study, Amarantaceae. The circumscription of Amarantaceae family has been considered in two forms. On the one hand, recent studies indicate that Amaranthaceae, including Chenopodiaceae, form a monophyletic lineage within the Caryophyllales based on morphology and molecular data. Amaranthaceae sensu lato includes Chenopodiaceae. However, after more investigations, it became apparent that the relationships between Amaranthaceae and Chenopodiaceae are still unresolved. They are now collectively referred to as the Chenopodiaceae-Amaranthaceae alliance. The main reason, as you can see in this slide, is because depending on the molecular marker, Amaranthaceae results as a monophyletic group, or it can be paraphyletic, like in the first phylogeny. You can see Amaranthus and Amaranthoideae and Gomphrenoideae that belongs to the Amaranthaceae family, and Chenopodiaceae that is lumped together with Amaranthaceae. So for that reason, it has been considered Amaranthaceae and Chenopodiaceae as an alliance. As you can see here in this slide, there were other molecular markers that were used to test the monophyly of Amaranthaceae, and most of the authors prefer to recognize Amaranthaceae in a sensu stricto, in a more reduced concept. The Angiosperm Phylogeny Group proposed to apply the name Amaranthaceae to the complete Amaranthaceae-Chenopodiaceae alliance. The broad family circumscription was also adopted in subsequent versions of the APG classification. However, since recent phylogenetic analysis rather indicate the monophyly of the core Chenopodiaceae, but are not yet conclusive about the position of the subfamily Polycnemoideae, the widely used family Chenopodiaceae is maintained. So, on the other hand, traditional systems of classification based on morphology, Amaranthaceae sensu stricto has been considered a separate family from Chenopodiaceae. So now I want to show you this slide. When I read the brochure that you kindly allow me to read, I really love this paragraph, this one, the first one. I thought that I was the only one with this experience. It's totally true that some journals emphatically emphasize in its guidelines for authors that they publish the findings in the light of the APG classification. One of these journals is Botanical Journal of the Linnean Society. We have a hard time convincing editor-in-chief that APG findings are not conclusive, and we found at the end a way to get the acceptance of our point of view. We are a group of taxonomists in the Caryophyllales group, and the leader of our team of work, as well the people involved, have dedicated plenty of effort in Amaranthaceae and Chenopodiaceae researches that doesn't match with APG classification. Okay, so Amaranthaceae, in a sensu stricto, the one that we recognize, is a cosmopolitan family with about 70 genera and 900 species. The centers of diversity for the family are in the North American deserts, the Neotropics, tropical, and Southern Africa and Australia. The family has been divided into subfamilies. Subfamily Gomphrenoideae with main distribution in America, and subfamily Amaranthoideae in the Old World. This is in this slide I present the flower structures of the most common member of the family. This is Amaranthus. The family Amaranthaceae includes some ornamental genera such as Gomphrena and Celosia for having colorful inflorescences, as well as Amaranthus crentus. The family includes Amaranthus as the most well-known and economically important member. Seeds of Amaranthus species have been used as pseudo-cereal grains. Here you can see that the black seed belongs to the wild species and the white seed belongs to the cultivated species. This species has been domesticated for the American Indian agriculture. Nutritional analysis show the grain to be comparable to true cereals as a carbohydrate source and superior in protein and fat content. We eat here in Mexico in a kind of cookie or a kind of candy. So there were too many classifications that were proposed within Amaranthaceae sensu stricto, based mainly on flower structures. You can see in this slide at least 5 classifications proposed along the time, since 1849 to 1993. These authors, well, depending on the author, recognize tribes or subfamilies or subtribes. Most recent classification of the Amaranthaceae proposed by Townsend in 1993 recognized 2 subfamilies based on the number of anther locules. The subfamily Amaranthoideae characterizes for having tetralocular anthers, whereas Gomphrenoideae has bilocular anthers. Each subfamily was divided into tribes and subtribes. So please remember this information. This is an example of corroboration test. As you can see in this slide, we only have hypotheses suggested by different authors on different times that used a group of characters that were considered diagnostic. They didn't have the tools and infrastructure that we have nowadays. Can you imagine how difficult it was for these naturalists to work without any of the devices that we have today, such as powerful microscopes, thermocycles, sequencers, even concepts have evolved, and they will continue as it happens with the homology concept. On the light of molecular data, subfamily Gomphrenoideae was tested as a monophyletic group based on RBCL and MATK data. In this study, the authors only included several genera. As you can see, there is one monophyletic group that includes Alternanthera, Tylestromia, Plutaparon, Gomphrena, Gilliminia, Frolichia, again, Gomphrena, Ivanti, Pseudoplantago. Iresine is also part of the subfamily Gomphrenoideae, but it's not resolved. So the well-defined monophyletic group was named the core Gomphrenoideae. The subfamily Gomphrenoideae has 19 genera and almost 400 species, mostly occurring in the New World. Recent studies of 9 genera Gomphrenoideae based on RBCL and MATK sequence data with representative taxa sampling of Amaranthoideae and Chenopodiaceae tested the monophyly of subfamily Gomphrenoideae. However, results suggested that it was unresolved. Additionally, classifications have placed Iresine in the subfamily Gomphrenoideae based on its two locular anthers. The results were inconclusive regarding the placement of Iresine in either the clade that includes most of the genera traditionally placed in the subfamily Gomphrenoideae or the amarantoids using both parsimony and Bayesian analysis. The importance of these previous molecular studies is that the authors found a well-supported claim named the core Gomphrenoideae, which didn't include the genus Iresine. And this, as you can see, this group was supported by a synapomorphy, which is the pollen type. This kind of pollen is unique. It is named a metareticulate pollen. As you can see, this phylogeny shows groups and ranks proposed in an informal way, like Amarantoids 1, Amarantoids 2, but also recognize some categories that were proposed in the past, like Gomphrenoideae or Amaranthi, for example. So this concept, this frame, reflects the author's criteria to define groups when they have named it in this way each clade. Although many members of the Amarantaceae family were included in this study, some questions remain unanswered, and it was still necessary to increase the sampling in order to better test the monophily of the subfamily Gomphrenoideae. This is another interesting concept about homology, as you can see here in this slide, which is related to pollen character. So in this slide, we have an example. As you can see, orthologs are structures with the same origin at the organismal level, whereas analogs are structures with different origins at the organism level irrespective of their phenotype or function. For example, on the left side, we have a metareticulate pollen. It means that we have a tectum and there is a pore or a hole in the middle. On the right side, we have a reticulate pollen. And probably you were thinking that it's the same condition. However, it's only an ornamentation form, and it doesn't mean that necessarily exists a pore or a hole in connection to this reticula. There can be less holes than reticulate structures. And in a reticulate pollen, we have the correlation of a net with a hole. So these pollen grades are not the same. The origin is totally different. Here, the character refers to pollen type, and the state of character, just to make it simple, we have two states, non-metareticulate and metareticulate. It's interesting to emphasize about this topic because in the past, it was mentioned that the kind of pollen in the Amarantaceae family was reticulate. However, after several studies, just recently was found that the correct type of pollen is metareticulate. We used two markers for DNA studies. We used the RNLF region, which is located in the large single-copy region of the chloroplast genome. The tRNL intron and tRNL spicer have become some of the most widely used chloroplast markers for phylogenetic analysis in vascular plants. The tRNLF marker was considered a good candidate region to resolve phylogenetic relationships with subfamily Gomphrenoideae. The intron in the chloroplast gene encoding ribosomal protein 16 is called the RPL16 intron. It is located in the large single copy region of the chloroplasts. The RPL16 intron has been found to resolve phylogenetic relationships at the infragenetic and familial levels. In addition, these were the first studies using these genomic regions in the systematics of Amaranthaceae. So you can see in this slide a typical alignment using as many sequences that we generated by that time in this study. So here the homology hypothesis in molecular data is determined by the same position of amino acid or mononucleotide within a sequence at the molecular level. For some reasons, these spaces in the DNA alignment suggest that some amino acids are either lost or gained, and those spaces are called indels. Here we had two big indels. All the species that we got sequences of Alternanthera share a common indel with a length of 300 base pairs. And that is a distinctiveness at this level for that genus. Whereas Iresine, Irenella, and Welleria have an indel of 316 base pair length. So after we conducted our molecular data and phylogenetic analysis, this is the consensus tree obtained from a most parsimonious tree, obtained with combined data from RPL16 and TRNLF. We found that Gomphrenoideae is monophyletic, and if you remember, it was difficult it was unresolved, the phylogenetic position of the genus Iresine. But based on new evidence and a major sampling, Iresine is part of the subfamily Gomphrenoideae. And this is exactly what I wanted to show you related to the corroboration tests. Maybe you remember that I presented several traditional classifications proposed in the past, and we wanted to test them. We wanted to know what kind of those three tribals or subtribals groups can be recognized once we conducted a phylogenetic analysis. So you can see that Trigomphrenae is not monophyletic, because Trizooplantagia is lumped in the whole Trigomphrenae. You can see that subtribe groups cannot be recognized either, because they are jumping along the phylogeny. So this means that these proposals don't pass a corroboration test. On light of new evidence, we proposed three major clades, and we name it in a very informal way. We name it the Gomphrenae clade, Alternantheroid clade, and the Iresinoid clade. Each one was supported with a high statistical support. Subfamily Gomphrenae share the synapomorphy for having bisporangiate anthers, and subfamily Amaranthoideae for having tetrasporangiate anthers. You can see in this slide that Gomphrenae clade can be characterized for having a very particular type of pollen grain. This is called Gomphrena type. And also you can see that Gomphrena is not monophyletic. Pseudoplantago has a very peculiar pollen grain. It is called pseudoplantago type. Alternantheroid clade is monophyletic. It includes Tyrostromia pedersenia and Alternanthera. And it has been characterized for sharing two kind of pollen types, Tyrostromia type and Pfaffia type. And this is the iresinoid clade. You can see that includes 3 genera, Iresine, Irenella, and Welleria. And the pollen type has been called Iresine type. So, how molecular data has helped to circumscribe groups and establish their classification? Well, hopefully I reached the point. Once we run a phylogeny, we can test traditional classifications and we can see if those tribes or subtribes can maintain on the light of a new phylogeny. It's very recommended, very desirable goal in plant systematics to obtain monophyletic groups. However, it's not always possible. So in those cases that we can find nested groups, like in the Iresine clade that share the presence of 2 other genera, so that means that it's recommended to make changes in the nomenclature of this group. And that's what it happens just recently. Irenella and Welleria now are synonyms of the genus Iresine, based on molecular data and pollen morphology. So as a conclusions for this part, I want to say that molecular data were used to corroborate hypotheses suggested in the past by taxonomists. Traditional taxonomy is valuable. Years of taxonomic observations with limited infrastructure and concepts. So groups were named based on monophily. Nested groups require changes in nomenclature. New proposal can be generated based on the results obtained. Phylogenies should be treated as hypothesis subject to be tested and susceptible to be updated. The last topic that I want to present is related with my studies conducted with ontogeny and DNA sequence in Amaranthaceae family. So this is a project that was developed as part of my PhD dissertation, and I was able to continue this study in collaboration with Dr. Alexander Brektas from Belgium. I choose some floral characters based on my previous studies in the subfamily Gomphrenoideae. The Gynoecium and Endoecium supply many characters considered to be important at the genetic level in traditional classifications, but they have never been studied in detail or analyzed in a phylogenetic context. And this study examined stigmas, anthers, pollen, and pseudostaminodia, a grown term, morphology, as well as evolutionary development of characters through the use of SEM, scanning electron microscopy. This is a typical flower in Amaranthaceae. Flowers in Amaranthaceae are very, very tiny. In fact, the bigger structures are the inflorescences. Each flower is subtended by 1 bract and 2 bracteoles, 5 tepals, according to the literature. And tepals is the most followed term, but it's also wrongly used. Here you can see the gynoecium and the androecium. The androecium is a very peculiar structure. You can see how the 5 stamens are united in the base, forming a staminal tube. You can see these appendages alternating with stamens. These appendages are wrongly named pseudostaminodia. So now, based on ontogenetic evidence, the tepals should be called sepals. This part of the androecium should be the androecial tube, and those appendages should be called androecial tube appendages. I was studying several genera, and I saw a lot of variation related to these structures. For example, in Blutaparon, these appendages are absent. In Centrostachys, they can be fimbrate. In Charpinteria, they can be entire or lobate and very tiny structures. In Gomphrena, it can be large and lanceolate. Lanceolate again. In Irisine, they can be triangular and like papillae around the tissue. They can be oblong or crenate or cordate. This study was interested to test a liaison hypothesis. A liaison is the first to suggest in evolution within the pseudostaminodia. A liaison first hypothesis suggested division and fusion of pseudostaminodia and the extent and type of fusion of these parts. He considered that perhaps 2 apical filament appendages in Gomphrena were fused along most of their length that would result in a structure reminiscent of the pseudostaminodium that occurs in Pseudogomphrena. An additional fusion could give rise to the pseudostaminodium present in Frolichia. The second trend of evolution suggested a progression in the reduction of filament size, increase in pseudostaminodia size, and fusion of pseudostaminodia with filaments forming staminal tubes. A liaison considered from pseudostaminodia of Alternanthera, the least advanced types in the family, derived the pseudostaminodia of Reliquiella. Reduction of filament length and fusion of pseudostaminodia with filaments would result in a staminal tube similar to that in Frolichia or Pseudogomphrena. Pseudostaminodia of Gomphrena are forked at the apex, deeper division, and decrease in a connection distance between the pseudostaminodia will result in a staminal tube similar to that in Gomphrena. We found 2 problematic conditions related to a liaison hypothesis. One difficulty was the controversial hypothesis of homology suggested for the stem tube of Gomphrena, and the second about the staminal structure of Phaphia. We didn't know how was developed this structure in the flower for this genus. So Brektas in 2014 examined the ontogeny of Irisine and Tydestromia. In this slide, you can see these 4 pictures of Irisine, and the remaining pictures belongs to Tydestromia. In this slide, you can see the flower development of Tydestromia, Tenella, and Irisine diffusa. Here you can see those primordia that will become on stamens, those on sepals. Here you can see primordium the gynoecium. And the second B figure, you can see how the ovary covers the ovule. In C figure, you can see a major older flower development. Here in the D figure, you can see some small primordia, and these primordia are alternating with filaments. The primordia will form an appendage that is called wrongly pseudostaminodia. But now we know that the correct name is appendages on the androecial tube. It's the same situation in Iresine. Those structures are bigger developed here in Alternanthera flavescens. You can see how big are these appendages, how big and how fimbriated they are. This slide shows the flower development of Alternanthera flavescens. Again, you can see different stages of the flower development. Here you see in figure D, primordias alternated with filaments, and these primordials are increasing in size. They became fimbriated and bigger. Part of my studies with Dr. Alexander Bregdas were to understand the ontogeny of this androecial tube for Gomphrena. Here you can see again a flower development of Gomphrena serrata, different stages, primordia that will develop on stamens and sepals, the gynoecium, the ovary covering the ovule. And it's interesting to notice that once the filament is formed, they develop tiny, tiny appendages on the filaments. So these appendages are not coming from a primordia located between two stamens. These filaments are developing from the filament. In this sense, appendages on the filaments and appendages on the staminal tube are not homologous, are not coming from the same origin. This is Phaphia, and here we're showing flower development of Phaphia fruticulosa. You can see again how these appendages are developing from the filament, and they became fimbriate. Okay, so we can say that appendages on the filaments seems to be the most evolved character related to appendages on the androecial tube or absent in the, as we can see in this phylogeny for the Gomphrenaoideae family. We also test, once we test the Liaison Hypothesis, we found them artificial. There is no way to support Liaison's ideas. Unfortunately, we don't have too much information about pollination types in the Amaranthaceae family. It has been hypothesized that Amaranthaceae pollinates by air, but there are very, very limited studies just watching possible insect pollinators in this group of plants. So we need more ecological observations related to pollination in Amaranthaceae. So conclusions, I can say that molecular data were useful to generate phylogeny, which was the framework to test Liaison's hypothesis, as well as character evolution. One more time, we have hypotheses suggested by a taxonomist that mainly conducted alpha taxonomy. Now we can test them or corroborate the hypothesis using modern tools. I hope you can see that these observations are very valuable. This is an example of evolution concept without using phylogenies. If liaison were recognized authority, then this can be called an authoritarianism in the family, which means an idea of evolution concept based on authority in the group. Ontogeny was the most important tool to test putative homologies. The use of a correct terminology impacts in the taxonomic descriptions and the alpha taxonomy. So now you shouldn't use the term pseudostaminodia anymore in your species of Amaranthaceae descriptions. New terminology was proposed in order to show a better description associated to structure, origin, and history. So finally, as general conclusions, I have only two main ideas to share. There exists a large amount of data generated, as well as classifications that are updated thanks to new information obtained by taxonomists. The value of those works is important since they allow us to re-evaluate, consider, or even use potential characters under the criterion of using them as possible hypotheses of homology. That is, we are still in the stage of analysis and data collection to be used by phylogenetic systematics. The never-ending work in systematics can be defined as a permanent review of previous and actual works in order to conduct new researches that verify or modify the actual knowledge in the light of the new information obtained. The success of a project for a taxonomist is based in knowing that there is not an absolute truth to prove. Each well-defined hypothesis or proposal carefully carried out in the herbarium, in the fieldwork, or in the lab will help to clarify relationships between species that allow us to better understand not only their differences, but why do they occur. Thank you for your patience and kind attentions.
The session is now open for discussion. You can ask questions.
Okay, so now I'm in the chat. And it seems that there are several questions. Okay. Or one of online not available. Is there any feedback from?
We have a question from Vinod, ma'am.
I,
He has asked for any fossil in the group and why is this group very complex and related to the biology geographical changes.
sorry, I cannot hear you.
Vinod has asked a question, ma'am. He has asked that why is this group very complex and is it related to any biogeographical changes?
Why is this group so difficult and phylogeography?
And he has asked if the complexity is because of any biogeographical changes.
Oh, okay. So it has been very difficult based on molecules. It seems that based on morphology, it has been very easy to differentiate Amaranthaceae from Chenopodiaceae, for example. They share too many morphological characters. So people really interested, we're a few taxonomists that use molecules, and we believe that these two families should be separated and should keep like different families. So I think that we need to get much more data and much more sampling. For example, the leader of this group, which is Dr. Thomas Porsche, who is the director of the botanical garden in Germany, he's the one that is developing a lot of research at genera level. As you can see in the phylogenies, there are too many paraphyletic groups. So he's trying to increase the sampling, molecular tools, morphology. So for that reason, these taxonomic groups sometimes are very difficult because flowers are so tiny, 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 are really small, and sometimes the differences depends on measurements. How big is the flower? How short is the flower? So sometimes it's a nightmare to define a species in this group of plants. And about biogeography, I have seen some phylogenists that have actually used the molecular data that I have generated. And there are some interesting hypotheses using photosynthetic pathways and a rank of humidity, of aridity, or semi-humid conditions. Just recently in 2019, 2018, there is a paper that came out talking about gonfrenoidy and biography and environmental conditions. But the people are really very interested about photosynthetic pathway, basically. There are too many C4s and C4C3s group of plants. Hopefully I respond to the question, I don't know. Jello, somebody?
Can I ask?
Yes. Yes, hello.
Hello.
Yes.
Can I ask you a question?
Sure.
Hello, ma'am. Myself, Dr. Suresh from Kerala. And first of all, I would like to appreciate you for this excellent presentation. It's very informative.
Oh, thank you very much.
My question is like, for this evo-devo, like evolutionary developmental biology, people has been using various genes like Madas box or something,
Exactly.
that kind of transcription factors that involved in floral development. So how that kind of gene is showing its usefulness in the Amaranthaceae family or something related to the Caryophyllales group?
I love your question. I really love it.
Thank you.
I really love it because as you can see, I'm traditional taxonomist.
Okay.
And I know a little bit about molecular data, but now phylogenomics and evo-devo tools is like to start another PhD, and I had to reborn again. I became an evo-devo scientist, right? So I have a collaborator here in Mexico. He's a specialist in evo-devo. And once we found this structure, once we found these new organs, we want to test if this is part of the androecium or part of what part of the flower structure in this matbox model are those structures. So we started this project. We have had very good results, but we need money because we need to get transcriptomes to design the primers for some regions in order to get the other matbox genes amplification. So we're in the middle of this project, but there's nothing. Actually, there is only one manuscript written by a British guy. I'm sorry, I forgot his name, but he was testing matbox related to sepals and stamens in Caryophyllales group, but I don't remember exactly which one.
Okay. Okay.
That has been the only study that I know. But in our topic, this is now our new goal, to
Okay.
study mad bugs for the new organs. But usually there are not too many projects focused on that research.
Okay.
So
Okay.
I really love your question, because I really would like to develop that topic.
Thank you, ma'am. Thank you.
Hello.
Yes.
May I ask you one question, Dr. Ivonne? I am Dr. Anil Kumar.
Oh, Dr. Anil, finally I know you.
Yeah. Okay.
Thank you very
See,
much
my
for your kind invitation. Thank you so much.
okay. My question is, I believe still there are two species of Alternanthera, that is the Ficoidea and Tenella.
Yes.
See, what's your opinion about the species status of these two?
Yes.
Is it really possible for us to distinguish?
Yes. This is a very tough question, because I had seen a variety material, and I had seen that it has been very difficult to recognize them as a species. In fact, I agree to recognize them as a subspecies. It's I think that we have to follow that classification. It's very, very difficult. So I have to work on the Mesoamerican flora, and I think that I will follow the recognition of Tenella and Ficoidea as subspecies.
Okay, okay. Okay, thank you, ma'am. Thank you.
But your question is very interesting, because we need to conduct molecular data that will help us a lot for a better definition, and maybe at population level, to get a lot of sequences of whatever is the concept of Tenella and Ficoidea, and included in a bigger picture, in a bigger framework of Alternanthera phylogeny. So that will help us a lot.
Yeah. Okay, thank you.
Sure, Professor.
Okay, I want to ask a question. Ma'am, myself.
Yeah. Finally, I know you.
First of all, I want to say that it was a very nice presentation, very informative. Myself, working under Dr. Anil sir, working in this taxa, I have a few doubts.
Sure.
I want to ask you that whether the variation in pollen morphology and seed surface architecture is consistent to demarcate at species level. Whether at species level, the variations of pollen morphology.
No, it's not possible. It seems that I have seen pollen morphology at different levels. I have been studying at subfamily, genera, subspecies, and I have talked about this topic to this professor, Thomas Borsch, and we agree that pollen morphology is very, very, very different at subfamily level or at major level. However, I have to say, that once I studied Tidestromia, I found a species that the morphology was very, very difficult. But I was suspicious that this species should be divided into subspecies. So I was very surprised when I studied pollen architecture, because one pollen was silent and the other pollen had spines. Yeah. And that happens at the infraspecific level. And that was the first time that I saw this condition within Amaranthaceae family. And it's very rare, it's very weird, because pollen is supposed to be different in major levels. So I'm not sure about it, because that helped me a lot to recognize species, really species within the genus. Maybe I was lucky, but the common knowledge is that pollen architecture should be at major levels.
Yeah.
Yes. For example, we have measured a lot of pollen grains, and maybe you can get few characters measuring sizes of spines, of pores, tectum, but it's not very informative. But that's my experience with pollen.
Okay, ma'am. Thank you.
Sure.
And there are a few questions in the comment box. I can read a few. Srija Rajiv has asked about the adaptability of Amaranthaceae members. Amaranthaceae members are fast spreading and they are very adaptable. Are there any molecular studies behind this adaptability and fast spreading behavior of Amaranthaceae members?
Sorry. Can you repeat your question? It's because the voice is cutting.
Okay, I shall repeat. The Amaranthaceae members are known for their adaptability and fast spreading behavior. So, is there any molecular studies which can throw some light into the adaptability and fast spreading nature of Amaranthaceae members? Srija Rajiv has asked the question.
Is it possible that you can write it for me, please?
Yeah,
the
I
sound,
can.
The sound goes and by.
Yeah, I can write this in the
Yes,
chat
please.
box.
Yes, thank you, because I cannot hear you well.
Okay.
Like ornamentals, please. What about the molecules? Can you say about the adaptability of fermentation members? Facts. Adaptability of fermentation members. What can I say about it? We have found some genera that are adapted to gypsum and gypsum soils, for example, very arid environments. Most of them are tropical. In the light of evolution, I know that there are only 2 genera that show very dramatic conditions to be adapted for these arid environments. I don't know more about other adaptability conditions for environment. And if about alternative, a project which is related to some species from the Galapagos. Since that there are very specific endemics in the Galapagos, and we are trying to understand the morphology, taxonomy, and to understand how this group of plants radiated in the islands. But I don't know more about the adaptation in this group of plants on the land. There's another question. Do you have any fossil evidence and how they are evolved the phylogenetic trees, since many of the species paraphyletic? If you date in the tree, it will be quite old diversifications. I haven't used molecular clock. I haven't used fossils. I know there are some papers that I have used, basically pollen. They have this kind of information about pollen fossils. There are very few groups, few phylogenies. So, for example, the groups that I study, like Tidestromia, there are no evidence about any kind of fossil. I should use the assumptions of the program if I want to do this kind of dating, the phylogenies. And maybe if we are talking about Amaranthaceae sensulato, I'm sure there are other options to calibrate the phylogenies, to use more evidence related to other kind of fossils. But no, I'm not an expert on this topic. Okay, you have a city, okay.
Okay, thank you very much, ma'am, for your response.
Excuse me?
Thank you very much, ma'am, for your response.
Okay. Okay. Well, thank you so much to everybody. Thank you. I really want to thank to Dr. Anil Kumar and to Arya for being so patient, for invitation. And hopefully, once the pandemic conditions are over, you can come to visit us here in Mexico. Hopefully we can make an interchange of students, or we can collaborate for future projects. You are always welcome to come to Mexico. Thank you very much.
See, ma'am, I would like to say a few words
Thank
on
you.
vote
Thank
of
you.
thanks. I would like to say a few words for vote of thanks. And as I understood, only few
Sure.
people, as I understood, only few people work on the Amaranthaceae family. One is yourself, I know, and there is Dielio Yamonico, with whom also we have some kind of collaboration, and Dr. Liaison, Dr. Emmanuel Guaccio. And
Okay.
you have your presentation was extremely useful, very informative. And I hope and I believe that most of the researchers, they might have got an idea about the phylogenetic concepts and the molecular tools that can be applied in the taxonomic realm. And
Sure.
when I invited, without any hesitation, you accepted our invitation. I would like to express special thanks for you, ma'am. Thank you. Thank you very much.
Thank you so much. So, good night, Mexico. Good morning, India.
Yeah. Thank you. Okay.
Okay. Thank you so much.
Okay.
Bye-bye. Bye-bye.
Bye, ma'am.
Bye-bye.